Welcome, dear students, to this enriching journey exploring how unspecialized cells become specialized, their roles in development, tissue maintenance, levels of potency, regulatory mechanisms, influencing factors, and groundbreaking applications in stem cell therapy, regenerative medicine, cancer biology, tissue engineering, and disease modeling. Yes, you're right. We are going to discuss about basic principles and characteristics of cell differentiation. So, learning objectives for today's discussion are: we'll be describing how unspecialized cells develop into specialized cells and discuss its role in development, tissue maintenance, and repair in multicellular organisms. We'll differentiate between the levels of cell potency. We will describe the stages and regulatory mechanisms of cell differentiation. Analyze intrinsic and extrinsic factors influencing cell fate. Last but not the least, we will identify and evaluate applications of cell differentiation. Cell differentiation is the process by which unspecialized cells, such as stem cells, develop into specialized cells with distinct structures and functions. It enables a single fertilized egg, zygote, to give rise to the diverse cell types that form tissues and organs in multicellular organisms. Early in development, embryonic stem cells possess the potential to become any cell type, while some stem cell populations persist in adult tissues. Differentiation is driven by tightly regulated changes in gene expression influenced by genetic and environmental cues, leading to variation in protein production, shape, function, and cell interaction. Specialized cells such as muscle cell for contraction, neurons for signal transmission, and red blood cells for oxygen transport generally retain their identity. However, advances in reprogramming have shown that some differentiated cells can revert to a pluripotent state, producing induced pluripotent stem cells, with applications in regenerative medicine and disease modeling. Coming to basic principles of cell differentiation. It occurs through regulated gene expression influenced by genetic programs and environmental cues, such as hormones, transcription factors, and signaling molecules. As cells specialize, they gain distinct structures and functions. For example, red blood cells lose their nucleus for hemoglobin storage, while neurons extend exons for communication. Differential gene expression. Despite sharing the same genome, cells differ in function due to selective gene expression, where only certain genes are activated based on developmental cues. This produces specialized proteins, organelles, and pathways. For example, muscle cells make actin and myosin for contraction, while pancreatic beta cells produce insulin, making gene regulation central to cellular diversity. Cell potency and specialization. Cell potency refers to a cell's ability to differentiate into other cell types. As development progresses, cell potency becomes increasingly restricted. Totipotent cells, such as the fertilized zygote, can generate all embryonic and extra-embryonic tissues. Pluripotent cells, for example embryonic stem cells, can differentiate into any cell from the three primary germ layers, that is ectoderm, mesoderm, and endoderm. Multipotent stem cells, They're limited to forming a range of cells within a particular lineage, such as blood cells from hematopoietic stem cells. Oligopotent and unipotent cells have progressively narrower differentiation potential, leading to a few or single specific types, respectively. This hierarchical loss of potency ensures tissue specificity and proper organismal function. Stages of differentiation. Cell differentiation occurs in several recognizable stages. Number 1 is the stem cell stage. These are undifferentiated cells capable of self-renewal and broad differentiation potential. Progenitor cell stage. These cells are partially differentiated, committed to a specific lineage, and undergo several rounds of division before further specialization. Terminal differentiation stage. Cells reach their final form, acquiring a fixed structure and function. These cells typically exit the cell cycle and stop dividing. Number 4 is regulatory mechanisms. Their students' multiple molecular systems control and stabilize the differentiation process. Gene regulatory networks consists of interacting transcription factors and signaling proteins that determine gene expression patterns. Transcription factors are DNA binding proteins that either activate or repress genes crucial to specific cell fates. Epigenetic modifications such as DNA methylation and histone modification influence chromatin structure and gene accessibility without altering the DNA sequence. Signaling pathways, for example WNT, Notch and Hedgehog, coordinate cellular responses to external signals, guiding lineage specification and morphogenesis. Asymmetric cell division results in two daughter cells with distinct destinies due to unequal distribution of cytoplasmic determinants or positional cues. These mechanisms ensure accuracy and stability in cell identity across tissues and developmental stages. Number 5 is the influence of microenvironment, niche we talk about. A cell's behavior and fate are not solely determined by intrinsic factors, but also by its surrounding, referred to as the cellular microenvironment or stem cell niche. Elements influence gene expression and guide differentiation. Growth factors and cytokines, cell-to-cell communication, mechanical forces, and extracellular matrix elasticity, oxygen concentration, and nutrient availability. Now, key characteristics of cell differentiation. Number 1, cell potency and developmental restriction. One of the core attributes of differentiation is cell potency, which refers to a cell's capacity to develop into different cell types. As I already told you, totipotent cells like the fertilized egg, pluripotent cells like embryonic stem cells, multipotent cells differentiate into limited range of cells within a specific lineage, or unipotent cells, which are restricted to producing only one cell type. Number 2, regulation of gene expression. Differentiation is driven by differential gene expression, where only one gene is required for a cell-specific role are activated and others repressed, producing specialized proteins. Number 3, role of epigenetic modifications. Beyond direct genetic control, epigenetic changes such as DNA methylation and histone modification also contribute to differentiation. These modifications affect gene accessibility and transcription without altering the DNA sequence. Importantly, these changes can be inherited during cell division, helping maintain the specialized identity of daughter cells. Number 4. Irreversibility and lineage commitment. Differentiation is often irreversible due to lineage commitment. Though advances like induced pluripotent stem cell technology show that some mature cells can be reprogrammed to regain a pluripotent state. Number 5. Responsiveness to inducing stimuli. Differentiation is triggered and directed by internal signals, for example, transcription factors, and external stimuli such as growth factors, hormones, cell-to-cell interactions, or mechanical cues. These stimuli initiate signaling cascades that led to alteration in gene expression and cell behavior. Number 6. Morphological and functional transformation. During differentiation, cells undergo visible and functional changes, such as ensuring cells are equipped to perform specialized roles. For example, neurons extend long axons for electrical communication, while muscle cells contain contractile proteins for movement. These include variations in shape and size, organelles and internal structures, membrane properties and receptor profiles, metabolic activity and response to stimuli. Number 7, dear students, stability and maintenance of differentiated state. Once a cell differentiates, its identity is generally stable and long-lasting, even if the original signals that induce differentiation are no longer present. This stability is maintained by epigenetic mechanisms and continuous expression of lineage-specific genes. Number 8. Plasticity in certain contexts. While most differentiated cells maintain a fixed identity, some exhibit plasticity, meaning they retain the potential to be reprogrammed under specific conditions. This plasticity is a crucial concept in regenerative medicine and developmental biology. Number 9. Functional specialization. A hallmark of differentiation is the emergence of specialized functions. Each differentiated cell type contributes to the organism by fulfilling a unique biological role. Muscle cells for contraction and movement, epithelial cells for forming protective barriers, neurons for signal transmission, immune cells for defense, blood cells for transport and immune surveillance. Types of these cell differentiation. Differentiation occurs in various contexts throughout life, and it can be categorized based on developmental stage and tissue environment. Number 1. Embryonic differentiation. This form of differentiation takes place during early development. Examples include the formation of ectoderm, gives rise to nervous system and skin. Mesoderm forms muscles, bones, and blood. Endoderm develops into the digestive and respiratory tracts. Postnatal and adult differentiation. In adult organisms, multipotent or unipotent stem cells continue to generate new specialized cells for growth, repair, and tissue homeostasis. For instance, skin cells, blood cells, and intestinal epithelial cells are continually replenished through differentiation. Induced differentiation. In laboratory and therapeutic contexts, scientists can direct stem cells or reprogram cells to differentiate into specific lineages under controlled conditions. This technique has immense applications in regenerative medicine, drug testing, and disease modeling. Types of cell differentiation post-embryonic and specialized pathways. The major types of cell differentiation beyond embryogenesis include somatic differentiation. This form of differentiation occurs after birth and is crucial for tissue renewal, repair, and regeneration throughout an organism's lifespan. In this process, multipotent or unipotent stem cells residing in adult tissues give rise to specific functional cell types. For example, hematopoietic stem cells located in the bone marrow continuously differentiate into various types of blood cells, including erythrocytes, leukocytes, and platelets. Dedifferentiation. Dedifferentiation refers to a reversal of the differentiation process, in which a specialized, mature cell reverts to a more primitive or less specialized state. Although rare in most vertebrates, this process is seen in certain organisms capable of regeneration. Example: in salamanders, cells at the site of limb amputation dedifferentiate to form a mass of progenitor-like cells, blastema, which then gives rise to the regenerated limb structure. Dedifferentiation is also studied in tissue engineering and cancer biology, where loss of specialization may contribute to tumor progression. Redifferentiation. Redifferentiation is the process in which dedifferentiated cells regain the specialized characteristics, sometimes re-entering their original cell fate or adopting a new one. Example: in plant tissue culture, dedifferentiated plant cells, callus, can redifferentiate into root, stem, or leaf tissue under the influence of specific hormones. In animals, this process is often observed during wound healing, where cells revert temporarily and then re-specialize to restore tissue function. Transdifferentiation. Transdifferentiation involves the direct conversion of one fully differentiated cell type into another, without passing through a pluripotent or stem-like state. Example: experimental studies have shown that pancreatic exocrine cells can be reprogrammed to behave like liver cells under the influence of specific transcription factors. This process has significant implications in regenerative medicine, where transdifferentiation could bypass the need for stem cells. Terminal differentiation. Terminal differentiation makes the final stage of specialization, where mature cells like red blood cells and neurons gain specialized functions, lose division ability, and rely on progenitor cells for replenishment when damaged or lost. Dear students, now let's talk about factors influencing cell differentiation: genetic structure and regulation. A cell's identity is shaped by gene structure, where sequences like promoters, enhancers, silencers regulate protein production. Mutation in these regulatory regions can alter gene expression, disrupt differentiation, and continue to developmental disorders or diseases such as cancer. Environmental factors. The external environment plays a vital role in influencing the differentiation of cells. Factors such as temperature, oxygen levels, nutrient availability, and chemical signals can affect gene activity and protein function. Oxygen supply can influence pathways like hypoxia-inducible factors, which in turn affects cell metabolism and fate decisions. Temperature variations and chemical exposures may disrupt signaling pathways or damage regulatory proteins, thereby altering cell behavior and developmental outcomes. Hormonal and chemical signals. Hormones and signaling molecules are among the most important extrinsic factors guiding cell differentiation. These substances act as messengers, transmitting signals from one cell to another and coordinating responses across tissues. Growth factors such as EGF or FGF bind to specific receptors on target cells and initiate intracellular signaling, cascades that influence gene expression. Hormones like thyroxine, estrogen, and cortisol plays a role in development and differentiation of various tissues, such as brain, muscle, and reproductive organs. Intercellular communication. Cell-to-cell interaction are central to ensuring that differentiation occurs in a coordinated and organized manner. Cells communicate using direct contact. Membrane-bound receptors are there, secreted factors, or through structures like gap junction, juxtacrine, and paracrine signaling, allows cells to influence their neighbors, ensuring synchronized differentiation within tissues. Influence of the cytoplasm. The cytoplasm of a cell contains organelles, signaling molecules, and proteins that can impact gene expression during early stages of development. In the zygote and early embryo, uneven distribution of cytoplasmic components during cell division can result in daughter cells receiving different regulatory molecules, thus guiding them down different differentiation paths. Developmental stages of the organism. The timing and developmental context of a cell plays a crucial role in determining its differentiation capacity. As the organism progresses from embryo to adult, the potency of stem cells become more restricted and differentiation signals change accordingly. Fetal tissues development, where pluripotent cells rapidly differentiate to form organ systems. Postnatal growth and repair, where multipotent stem cells are activated for maintenance and regeneration. Wound healing, where previously quiescent stem or progenitor cells are activated to replace lost tissues. Examples of cell differentiation in plants and animals. Cell differentiation is a universal process in multicellular organisms, enabling the development of specialized cell types from a single undifferentiated zygote. While the basic concept is conserved across kingdoms, the mechanisms and manifestations of differentiation differ in plants and animals. Cell differentiation in plants. Plants, like animals, begin their development from a single fertilized cell. This zygote undergoes a series of divisions to form an embryo. The development of plant embryos halts temporarily during seed dormancy, resuming upon favorable conditions. Once growth recommences, the embryo gives rise to specialized regions called meristems, which act as centers of continuous growth and differentiation. Cell differentiation in animals. In animals, the differentiation process begins immediately after fertilization. The zygote, initially totipotent, divides repeatedly, forming a blastocyst. Within the blastocyst, cells begin to specialize, transitioning from totipotent to pluripotent states. These pluripotent cells eventually give rise to all the tissues and organs of the body. Animal development is guided by three primary germ layers. The ectoderm forms the nervous system and skin. The mesoderm gives rise to muscles, bones, blood and connective tissue. The endoderm forms internal organs such as lungs and gastrointestinal tract. A key component of animal differentiation is the role of stem cells. These cells, which vary in their potency, include totipotent cells that can form all cell types, including extraembryonic tissues, pluripotent cells capable of generating all body cell types, but not the extraembryonic structures, multipotent cells found in adult tissues that differentiate into specific lineages, such as blood or skin cells. Coming to applications of cell differentiation in molecular biology and medical sciences. Cell differentiation possesses potential application in molecular biology and medical sciences. It has explored new areas of research in biological sciences and clinical practice. The key applications of cell differentiation are listed as stem cell therapy, cancer research, tissue engineering, organ regeneration, gene expression analysis, developmental biology, drug testing and toxicology, personalized medicine, disease modeling, and reproductive technology. Cell differentiation transforms unspecialized cells into specialized types, enabling growth, tissue organization, and lifelong function in plants and animals, guided by precise gene regulation and environmental cues. It underpins development and repair. Errors can cause disorders including cancer. Insights into this process drive breakthroughs in regenerative medicine, stem cell therapy, and tissue engineering, offering powerful strategies to understand, prevent, and treat complex diseases. Dear students, this is all for today. We'll get back to you with another lecture. Thank you.